Hello, everyone, and thanks for inviting me to give a talk about my PhD research. And recently, I graduated, and now I've started a postdoctoral position in Uppsala University. So, the title of my talk is PIP2 mediated oligomerization of endosomal sodium proton exchanger, which is NHE9. A little bit of introduction about what are sodium proton exchangers. So, basically, these are membrane proteins which mediate exchange of protons for sodium ions. So they are found in all kingdoms of life and they regulate intracellular pH, sodium levels, and cell volume. So these are basically antiporters which transport the substrate in a direction opposite to the driving ion. So in these are basically solute carrier transporters and they have an alternating access mechanism where the substrate is bound to the outer open state and then it transitions to an occluded state inside the membrane and then the substrate is transported inside the cell in an inward open position. And sodium proton exchanges, they follow an elevator model where the substrate only binds to the transporter domain. So one of the domain is static and the other domain is mobile, and the substrate binds to this mobile domain, and then there is a transition to an occluded state, and then it moves to an inward facing state where the substrate is transported inside the cell. So these exchanges can be electrogenic or electroneutral. So they can either transport two protons in exchange of one sodium ion, or they can exchange one proton in exchange of one sodium ion. So coming to the mammals, so the sodium proton exchanges that I'm sharing is a mammalian exchanger, and in mammals there are 13 different sodium proton exchangers. They are classified into SLC 9A, B, and C. So the SLC 9A are like exchangers which are again subclassified based on their localization. So some are localized to plasma membranes, which are from NHE1 to NHE5, and they have different functions inside the cell. And then there are intracellular NHEs, which are NHE6 to NHE9. And then there are SLC9B family, which is very close to the bacterial homologs. And then SLC9C, which is having an extra voltage sensing domains, which are similar to like ion channels. So they are like exchangers plus ion channels. So these mammalian exchangers, they have a and they have the dimer interface, and then there is a transporter domain, also called as core domain, which actually has a broken helices where the ion kind of binds to the this region, and then it is transitioned towards the inward open uh, state where the ion is transported inside the cell. The interesting part about these exchanges is they have very long C-terminal tail, and these tail is supposed to bind like some interacting partners such as carbonyl or CHIP1 or some lipids such as PIP2. One interesting thing about these intracellular exchanges is that they have like an extra loop domain, which is at it's above the scaffold, like the dimer domain. And these uh, like in the previous structures that we have done, we didn't see any density, like we couldn't resolve this region in the previous structure. So our aim was to explore what this loop domain is doing to the transporter. So these energies, like which are endosomal, they are actually localized to different compartments inside the cell. So if you see like NHE6 is localized to like early endosomes, then NHE9 is to like late and endosomes, and then NHE7 to like Golgi, and NH8 also to like trans Golgi region. And they have like different pH range. So each of these compartments has different pH, and this pH has to be regulated. So main role of these exchanges is to maintain this pH inside these organelles. And also like what we explored in the physiology, like these endosomal compartments have different pip distribution. So you will find like different pips localized to different endosomes. So if you see like in plasma membrane, you only find majorly like PI4, 5P2, whereas in case of early endosomes, you have PI3P, and in case of late endosome, you find PI3, 5P2. So we were trying to explore like what is the role of like lipids, and then what is the role of these NHEs. There is a difference of pH, so all these factors. And then we found, when we studied the structure of NH9 before, we could see that this loop domain was not resolved, and this had like a positively charged lysines at the interface. So we like, what, why these charged residues on this loop region, what they must be doing. And so we try to resolve the structure of NHE9 again with a loop domain. So for this, we try to make this exchanger a bit static. So we try to resolve it at an acidic pH, which can make it more like static. And then if the transport is less active, maybe we could capture the structure of the loop domain. So we try to do that. In some of the density, we could see like there is some flexible loop, but it was really hard to resolve. And then we selected like some particles which had this loop, and then we did a 3D variability analysis where we clustered these particles and then tried to resolve like this 100,000 particles further to get the structure for the loop. Yeah, we did see the loop. The resolution was not that good enough, but at least for the, like when we do the focus refinement for the loop, we could see like there is like a domain swapping between these regions. And then the AlphaFold came, and then we tried to build a model using the AlphaFold, and we could see like when you make an AlphaFold like dimer and we try to fit in the densities, you could see like this is domain swap. Like um, yeah, so like one of the regions of the loop domain is facing like close to the other protomer, so they are like flipped in the region. And we could interestingly see that the lysines that we found, which were there in the sequences, they are clustering towards the dimer interface. So it built us like a curiosity, what are these lysines and positively charged residues doing with the dimer interface. So further, we try to resolve the structure of NHE9 with the lipids. So for this, actually, we try to put some PIP lipids because they were known to function or regulate these exchanges. But unfortunately, we couldn't get any structure with these lipids. So what we did was we enhanced our purification with crude lipids, and this crude lipids picked up the necessary lipids which were at the which would be required to stabilize the dimer and also to the loop domain. So when we had this structure, we could see like there are some lipid densities at the dimer interface, and we could see that the positively charged like the lysines they cluster above these lipids. And initially we thought that the lipid would be the pi45p2. So we were trying to model that, but we could see like it it didn't show, like the density was more clear for pi35p2. And it also physiologically made sense because NHE9 is localized to late endosomes, and the major lipid found in late endosomes is pi35p2. So which all made sense. So to further confirm this, we did this assay which is like optimized in our lab. It's called as thermostability assay. We use a GFP purified fusion. We add lipids to it, and then we incubate it for like 30 minutes, and then we add a detergent, which is beta OG. So whatever protein is like aggregated, that can be precipitated using this detergent. We heat the sample at an increasing temperature, like from let's say 10 degrees to 80 degrees, and then we spin it for almost half an hour, and then we pull out the sample which is non-precipitated, like the uh, yeah, like the non-precipitated sample, and then we measured the fluorescence intensity for that. And we could see like when we measured the fluorescence intensity, the TM that we calculated for the purified fusion was around 51 degrees, but like if you add lipids to it, the TM shifted, and the new TM for with pi45p2 was around like 59 degrees, and for 35p2 was 66 degrees. So 35 was more like stabilizing compared to 45. And so that TM shift was around like 8 degrees for 45, and 16 15 degrees for pi35p2. Again, like what we do is like we take this sample again, and we can inject it on fsec to see the quantity of like monomer and dimer present in the sample. And we could see like PI35P2 was stabilizing the dimer compared to 45P2 and the other lipids such as POPC. So we could clearly see that the lipid which is preferred by this protein is PI35P2. Again, like to measure the activity for this transporter, we use this assay called a solid state membrane electrophysiology assay. And we make proteoliposomes first. So we take like crude lipids such as yeast polar, and then we add like detergent, membrane protein, and the liposome. We incubate for some time, and then we do a filtration, and then we get this membrane protein inserted into the liposome. So we get proteoliposome. We put it on a gold sensor, which I think was already discussed by Rosa, like how this solid state electrophysiology experiments work. So we did with the similar assay, we had the setup, everything. And then what you do is like you add a non-activating solution. So in this case, like sodium is the substrate for this protein. So you would take any ion like lithium, or like we use choline chloride, which has a similar charge as sodium, and but doesn't like it's not a substrate for the protein. So we add that as a non-activating solution. Then we add the substrate, which is like sodium, and then again a non-activating solution to have like an off signal for this. And in our case, like for these exchanges, we say that it's not calculating the actual km, but it's calculating the kd, like binding affinity, um, because even the binding of the substrate can drive a conformational change in the protein, and this change can also give a change in the membrane potential, which can be measured. So this gives like a transient current, but not the actual transport. So we calculated the binding affinities in the presence of this lipid. So what we do is like we incubate the protein with this lipid, and then we incorporate this whole sample into the liposome. So the lipid is already bound to the protein, and then we measure the signals for each of the samples. So when we measure the signal for the wild type without any lipid, we could see like the signal was uh, around 3, the peak currents that we got. And then when we added this PI35P2 lipid, like the signals were higher, like around 7.5. And then we also made a variant for this, which is a dead mutant, which we call, it's the mutant which is like it's the residues which bind the ion, and we mutated both of that. And then we measured the signal for this, and we could see like the signals were like very low. So we could say like this setup is now working fine. And then we measured the binding affinities for this. So the kd that we got for the wild type was around like 36 millimolar, but when we added this PI35P2 lipid, we could see that the kds were like around 9.3 millimolar. So which was like this lipid was enhancing the activity, or let's say binding affinity for the protein, for the ions. And then we also added PI3 and 4,5P2 lipids, and we could see like for with addition of 4,5, the binding affinities were around like 4.7 millimolar, which is very close to what we get for the wild type. So this lipid was not actually enhancing the activity of the protein. So to further confirm with our results, we did some MD simulations in collaboration with Lucy. So what we did was we put this lipid in at the dimer interface, and we tried to measure, like see if these lipids like make contacts with those positively charged lysines and the arginines around. And we could see like this, the lysine 105 was directly interacting. And when we mutated those lysines, we could see that um, there was no contact between like contact interaction happening between these residues. Then we also measured in the presence of 4,5P2, and it could clearly show that this protein preferred PI35P2 based on the contact probability. So it was very clear that this is the lipid which is binding to this protein. And again, like so as we know that PI45P2 was the major lipid at plasma membrane, so we try to express this protein at the plasma membrane and measure the activity of the transporter. And we could see like NHE9 did not have any activity at the plasma membrane, because there's no PI35P2 lipid present in the plasma membrane. So this protein is inactive when it's localized to plasma membrane. And then we measured the activity of the protein at the endosomes, and we could clearly see that there is a pH change when you have it expressed in the endosomal. Um, so this was also done in collaboration with Daniel Fuster. And from this, like, we build a model where we say that when NHE9 is expressed in the plasma membrane, there's no PI3-5P2. And this protein is very, like, this loop domain is very flexible. And because of that, like, there's no transporter is like less active. But when we express in endosomal membranes, you have this specific lipid and it binds to the dimer interface, stabilizes the protein, and then it also enhances the activity of the transport. And with this, I would like to conclude my talk. And I would like to thank all my lab members and my supervisor, David, for making me work with these exciting projects and giving me opportunity to explore about this lipids and membrane proteins in very